everyone. Welcome to a special SFN conference edition of Max Planck Florida's Neurotransmissions podcast. I'm actually joined today by producer Kevin. Hi, Kevin.
Hello. How are you doing?
I'm doing great. We've been talking to people, as you know, about their experiences at SFN, along with a very special research guest later in the podcast. But I wanted to take a minute and ask you about your experience at SFN. What do you think?
I think it's eye-opening to see the full neuroscience community in one room. I imagine for a lot of people when it's their first time, it can be quite overwhelming because you think about it and you recognize that there's all these different areas of neuroscience, but when you actually see an expansive room full of it, including vendors, including different training programs, including places like NIH and whatnot, there's a lot going on. You're meeting a lot of people. You're also reconnecting with people that you either met in a previous program or at previous SFNs. It's a great all-around experience.
It is. And it's also been fun talking to different people to sort of see everyone's unique perspective, depending particularly on their career stage, you know, how they feel about coming to a meeting like this sort of changes both with experience, coming here more than one time, but also as you progress in your career and you're looking to get different things out of a meeting like this. So it's been interesting. And we also get to talk to a clinical psychologist on the episode today, which was really great for me because, as you know, as a basic research institute, we don't often really get to talk about the actual difference that neuroscience is making in patients' lives. So that was really a wonderful opportunity for us. So without further ado, let's get to it. Enjoy. So I'd like to welcome a actual first-time SFN attendee, Alexis Green, who is a postbaccalaureate scholar at NIH and the National Eye Institute. Thank you so much for joining us on the podcast. We're thrilled to have you.
Thank you. It's lovely to be here.
Great. So as a first-time SFN attendee, it's been a while since it was my first time. So I'm just wanted to hear a little bit about your perspective on your experience when you first walked in the doors at SFN. What was your initial impression?
Well, I actually got here really early on the first day, and there was almost no one here. I only saw 100 people. So I thought that going to such a large convention center was not necessary. I think I got here at like 8:30, 9 o'clock in the morning, and I was like, oh, this isn't even that big of a deal. But then when it got to about 10:30, there were probably 5,000 people that I've seen, and I was like, oh, this is a really big conference. So I was like surprised that there were so many neuroscientists worldwide, and also surprised that they all came to this meeting in particular.
Yeah, so you've been here now for, 4 or 5 days now, right? So how did you spend most of your time here?
Most of my time I've spent in the lectures. The first day I had a poster and I presented that in the first session. So I was there. I got here early because I got a little lost. But I presented it from 1 to 5, and then I invited my mom and my brother so they saw me present.
Oh, nice.
And that was the whole first day. And then I went and I saw the exhibits for the art that people sell. And then after that, I attended the posters and talks for the people that are in my lab, and mostly attended lectures of interest.
Great. How did your poster go? What was it like having your first poster presentation here?
It was really busy, but I feel like I was well prepared because I went to a conference in May and presented, and there were a lot of people that are interested in my work and that just had a broad interest in like object recognition and identification. And but I find that the questions that I was asked here were more higher order processing and sort of reaction time based.
So maybe a slightly different perspective than you were getting before. Maybe you could tell us just a little bit about your research, what sort of the main question is that you're looking at.
Yeah, so broadly we focused on the neural mechanisms of vision and more specifically color and shape integration. It's more basic science. So, yeah.
Great. So one final thing I want to ask you is, now that you've been at SFN for a couple days, what would you tell maybe a first-time attendee who's coming? Do you have any advice or things you've learned having been through this experience?
Yeah, I would say try not to be overwhelmed, even though there are a lot of people and you'll hear a lot of different accents and languages and perspectives, but try to sort of immerse yourself in the experience. I mean, where else are you going to go where there's going to be 25,000 people doing the same thing that you're doing? And also try to take this opportunity, if you're a younger scientist, to network and maybe find a PI whose lab you're interested in and see their work, because when you're applying to grad school or medical school or trying to figure out what your path is, most of the research you do and the information you're taking in is strictly online, and you're not going to be in the same room as that person. But here you have the opportunity to speak to them directly, maybe speak to the people in their lab, and understand on a deeper level what they do in a way that relates to what you're interested in. And then also have fun, because you might meet someone who turns out to be your friend at a social. Like, I went to the Black and Neuro Social, and I don't know really any Black neuroscientists, so it was like really cool to experience that for me.
That's great. Yeah, I love the connection aspect of SFN. If there's something that comes to mind, I wouldn't mind asking you if there was either someone that you met or a particular lecture or poster that you went to that really got you excited.
Oh, actually, yeah. So downstairs, not downstairs, but like we are downstairs, but I found a poster here over there where they're focusing on non-invasive methods of working with animals. Specifically, I work with animals, and we're trying to find ways to work with them that don't involve any sort of like head posting or like permanent, I guess, restraints. So there was a poster where they did a 3D mask of a rhesus macaque, so that they don't have to give the monkey an implant. So the monkey just walks up to the mask to hold their head in place in order to track eye movements and sort of assess attention. And I thought that poster was really good, because now it can sort of like open the door to no longer have to give the monkeys head posts on the tops of their heads, so they don't have to undergo that procedure and have to recover in order to do science. Like, we can just use this more simple mechanism that takes longer to make because you have to make the mask, but it's less invasive, and it's really interesting to see in practice.
That's great. Yeah, better for the animal and better for the science too. So great. Thanks for sharing that. Well, thank you so much for joining us. We really appreciate it. It was great to hear your perspective. Thank you.
Thank you.
Welcome back. I'm here with Dr. Timothy Holford, a postdoc at MPFI in the lab of Dr. McLean Bolton, and we're so glad to have you, Tim.
Thank you. Yeah, super exciting.
First of all, I must say you look incredibly dapper. You're kind of showing me up here in my sweatshirt.
Yeah,
but
you can't see. Oh, wait, you can see.
You can see, actually.
Yes, I forgot.
Yeah,
oh my
we're
gosh,
on video.
Thank you.
No, you're looking really nice. I understand you have a poster presentation this afternoon.
My poster's this afternoon, so got to be ready for it.
Awesome. Do you want to give us a little sneak peek on what you're going to be presenting?
Oh, yeah, sure. So the poster's actually mostly based on thesis work that I did a few years ago, but then I kind of filled it out with some other couple extra controls and fill out the data set. Essentially, our lab studies neural circuit dysfunction in mouse models of autism and schizophrenia and those kinds of things. So I had an autism protein that we specifically knocked out in somatostatin neurons to get kind of a gauge of how a certain subclass of inhibitory neurons, those somatostatin neurons, how that actually contributes to the overall shape of the autism phenotype. So we looked at it behaviorally, found some phenotypes, not your classical total ASD with social and repetitive motion and all that, mostly just a fear and anxiety phenotype. And so we looked into the amygdala, as you might guess, a place that's known to regulate those kinds of emotional learning and that kind of fear and anxiety regulation. So we looked in there using a cool 2-photon circuit mapping single cell analysis technique to probe local circuit connectivity, and found that in these mutants there's just a drastic reduction in lateral inhibition within the central amygdala, and so they're not able to tamp down the inputs that are coming from the BLA and end up sending those further out, or at least that's the hypothesis, because there's a greater reduced inhibition there. It's resulting in these elevated levels of fear and anxiety. So that's kind of the short version of it, but yeah, it turned out pretty good. I'm pretty happy with it, so we'll see. Hopefully
That's
get
awesome.
some
I love when a
conversation.
surprise result kind of like shifts your direction a
Yeah,
little, and
yeah.
it's always exciting.
Yep, the flow of science.
Yeah. Well, I hope you get lots of people to your poster, although unfortunately it is Wednesday afternoon. Those of you who have been to SFN know that that's sort of the like worst draw you can get in terms of poster times.
Yeah, half the people have left already, and the other half are just as exhausted as I am.
Exactly, yeah. But how has your SFN been?
Man.
Do you come to SFN often?
I try to go, so I didn't go last year, right when they came back from the pandemic, but I try to go once every other year or so. I kind of switch off between SFN and then a smaller conference if I can. But yeah, I find it incredibly valuable. I really love SFN. And I actually went really early in my career, before, like right when I joined graduate school. I was still rotating through labs, and I was able to go to SFN, and it was just, it's really good to see just the incredible diversity of techniques that people use, things that they study, just the variety of approaches that people have, because, I had done a little bit of volunteer research in a small lab, before that, and I'm like, oh, this is what neuroscience is, and then I come here, and there's 30,000 people doing things completely differently, looking at approaches completely different ways. And so it really just kind of opens your eyes to the whole spectrum of neuroscience research. And I think that's really valuable to get early on in your career to see if you can incorporate some different ideas into your stuff. I think towards the middle of the graduate program, like when you're deep in the weeds of your research and you really need some specific feedback, some really, you want to see what other people are doing in your immediate field, that's when the smaller meetings really tend to bring you more value. But then towards the end of the arc as well, once you're getting close to finishing, sort of close to thesis time, looking for postdocs, looking for next steps, there's no better place to have 25,000 scientists all doing lots of different things. The number of connections that you can build, the types of conversations you can get into, it's really expansive for that kind of thing. So yeah, I really enjoy it. I understand there's some criticisms on like the scientific side of like, well, you can't get your questions asked, you can't have conversations with the big name people. And to a certain extent, that's a little bit true. But now, since the pandemic, like everything is videotaped. All the big lectures are on video, so I don't even go to those anymore. So I'll watch the large lectures later when we get back home, because we have still a couple of weeks access to those. And so I go to as many posters as I can, to the mini and nano symposia, and there you can get your questions asked. If you stay like 2 minutes after everybody rushes out of the room to go to the next thing, the people, they want to talk, they want to ask questions. So I've stayed behind and asked a couple of questions to some people and had, perfectly great access to them, been able to talk for a couple of minutes about voltage imaging with Adam Cohen and, stuff, simple things like that, they're there to help. They want to be helpful.
I'm always so impressed at how just approachable these scientists are. And there's no better meeting, where people from all over the world and past friends and future, potential connections can be made than SFN.
That's another thing. I mean, neuroscience is global, right? And so, especially being part of the Max Planck program, I've got friends in Germany, from when we were a partner institution, which now our graduate programs have diverged a little bit, but I still have plenty of friends from there. I know people who have moved on to have labs in Japan, in Italy, in Rome, and this is the place where everyone really comes together. So, it's cool. I get to catch up with my friends and see what they're doing. And not that you're busy, right? I mean, I haven't been able to read everyone's stuff from the papers they wrote or even if they haven't published stuff yet, like you can still check in on them. And it's always better to hear it from them anyway. So, yeah, I've had a great time reconnecting with some people and a short course that I went to. So I missed SFN last year because I went to a short course in Venice, like just kind of 2 weeks, really intense, more of a course though than a conference. Met a good group of people there, and several of them are here, so we've reconnected for that and went to the dance party together, and but yeah, SFN, it really shows the global side of neuroscience, which is incredibly helpful. Yeah, and then one other thing that I really like about SFN, in small meetings, you don't have as many vendors. Here, everyone who does anything about neuroscience is here. So earlier on in my career, when I was a graduate student, I wasn't necessarily the one responsible for all the equipment in the lab and when stuff was going wrong. But now, as a postdoc, like, it's almost my role is keeping all the microscopes functional and working and setting up new stuff. And so having access to every single vendor, all of them that are here, and if you think scientists want to talk to you, the vendors are dying for people to come and talk about their stuff. And so I've found that tremendously valuable. I've already solved problems. I've already got emails of things that we're going to, remote log into when we get back there. So yeah, they want to help and they're here for it. And they're all in the same place. And you don't have to track down 100 emails and, go through the weeds of all that. You just go talk to the person and say, hey, I like this, I'm interested in that, and they can point you in the right direction. So that's also really valuable.
A huge time saver.
Yeah, totally.
Awesome. Did you see any particular science that you thought was super cool?
Yeah, totally. So actually, two of the people that I was most impressed with were both part of this new SF Nova lecture series that they've started.
Yeah.
They're highlighting younger track scientists and getting a little bit earlier in their career. One of the guys, Ismail Abdou Sabour, he was who the postdocs had selected to come to our retreat. We were going to invite him to the retreat, but scheduling didn't work out. And so before that lecture, I walked into the lecture. He's standing there because he's getting ready to go talk. And I just went up and introduced myself. I was like, hey, we were excited to have it. I'm excited to see your talk. And he was super grateful and just said hi. We didn't talk for a long time, but it's just cool. I mean, they're there. They love science. They love people. They want to be involved. And then the other person as part of that lecture series, her name was Kanaka Rajan. She's doing really cool computational stuff, which is it's a field that I haven't really gotten into. I've been interested in. It looks cool, but I don't have a background there. And I don't know if you've ever read computational papers, but they can be a little difficult to really kind of get through. And so there's something about just hearing it from the person who's doing it, the way she was able to explain the process and what she's doing and why she's doing. It really just kind of solidified a lot of those ideas for me. And it was just really cool to see how she's using, these AI neural network models to try and model brain states and different behaviors. And, it's all cool things that I've been interested in, but really distilled down in a better way. So I went to her talk of that, and then she had a whole nother Meet the Scientist series where it was an expanded version of that with a lot more question and answer time. And yeah, it was really good. I found it really impressive to the point where I have my computer here, and in between some of the lectures I've went back and remote logged on, tried to run some of the simulations myself on my own data with limited success. Albeit it's going to take some practice, but I'm excited about some of those new horizons, new steps. And so, I mean, that's really the value of bringing multidisciplinary scientists together. You know, you wouldn't necessarily get that at a small circuits and, you know, proteins kind of version where it was just focused on what I was doing, you know. So branch out the techniques, you see different highlights. So it's been really cool.
That's awesome. Thank you so much for sharing your experience with us, and it sounds like it's been a phenomenal meeting for you.
Yeah, super cool. You know, we're having a great time. I've got my poster this afternoon, and then it'll be over.
Until next year.
Until next year. Yep,
All
we'll
right.
come back.
Well, good luck with the poster, and thanks so much for stopping by to talk to us.
Awesome. Thank you so much.
Yep. Bye.
Bye.
Welcome, everyone, to Max Planck Neurotransmissions. We are here at SFN with a wonderful guest today, Dr. Carolyn Rodriguez. Dr. Rodriguez is a psychiatrist and professor of psychiatry and behavioral sciences, as well as the director of translational therapeutics labs and associate dean of academic affairs at Stanford University School of Medicine. She has won many awards in her career, including the Presidential Early Career Award for scientists and engineers. And we're just absolutely thrilled to have her here. One of the things that I love most about SFN is that we really are able to see scientists across all areas of science, from the basic research all the way up to clinical researchers. And, you know, as a basic researchers, we don't often get a chance to talk to clinical researchers here on the podcast. So, Dr. Rodriguez, we are so thrilled to have you. Thank you so much for joining us on the podcast today.
It is absolutely my pleasure. It's wonderful to be here.
So before we dive into your research work, I wanted to actually ask you some sort of basic questions about obsessive compulsive disorder, which you have spent much of your career studying. And I had the opportunity to hear you speak this morning, and one thing that really struck me in your talk, but actually saddened me really deeply, was that you mentioned that people who suffer from OCD often spend 14 to 17 years before they seek treatment.
Yes.
Why do you think that is?
Yeah, absolutely. And thank you for saying that. It is devastating for patients to take so long for recognition of the community. And I think, it's hard to pinpoint exactly what it is, but what we think is it's a combination of things. So one is, and why I do a lot of sharing what are the signs and symptoms of OCD, the first one is recognition, right? So OCD is common, and it's characterized by obsessions that are intrusive thoughts, images, and urges that increase anxiety, and compulsions that are repetitive behaviors that serve to neutralize or decrease that anxiety. And there are examples in movies of people that think things are contaminated, and they will repeatedly wash their hands or take a long time having ritualized showers. But there are other types and themes with OCD that are a lot less common that can lead to misdiagnosis. So one of these is having intrusive thoughts of harm, feeling like you've run somebody over. Even though you know that you haven't, you can kind of put that together in your mind, but you have this feeling and anxiety. So that drives people to, look at the news, spend hours looking at the news, make sure that they haven't run somebody over, or driving back to the scene. And those are just a few examples where people can be misdiagnosed. The other is, I think is a real one, is just stigma. People being worried that they're having these thoughts. They don't know what's going on, and they're worried that something, that the doctor will misunderstand their symptoms. And so that's why it's important to seek out people, to have out there and have low barriers for people to reach out and seek help.
Yeah, and I was at least somewhat, felt a little better after I heard that, even with our current treatments, I think it's a pretty large proportion of patients that actually see some improvement of symptoms. What are the sort of current first-line treatments right now, and how effective are they? And then maybe we can talk about some of the limitations.
So we know that about half of people will get better with first-line evidence-based treatment for OCD. These include therapy called cognitive behavioral therapy with exposure and response prevention, and medications that are serotonin reuptake inhibitors. Those are things like sertraline and fluoxetine. And the therapy for OCD that is cognitive behavioral involves making a hierarchy of things that an individual is very fearful of, and then moving through the hierarchy in a progressive order, and trying to unlink the idea that you have to do repetitive behavior when you are exposed to the thing that you fear that is increasing anxiety. And we know that anxiety extinguishes over time, and so helping the person kind of see that they don't have to do those behaviors, and having a new form of learning that doesn't involve those repetitive behaviors is what we think that is helpful for patients with OCD. Yeah. So it gets a little bit even better than 50%, which is that if you then go on, if those things help a little bit but not enough, you can do augmenting things. So if you aren't SSRIs, you can add on exposure and response prevention and do even better. Or if you're on medications and you add on antipsychotic medications and augmentation, you can do even better. You can go on and try different SSRIs. So in all, if you go through the algorithm for the American Psychiatric Association, 7 out of 10 people will have reduction in their symptoms. So that is good news and cause for hope. The limitations is that not everybody gets better. So that's where my research comes in, to try and help those people who have tried the algorithm and are still having symptoms. And the other is that it can take a long time. So with SSRIs for depression, for example, in about 2 weeks you'll see if it has an effect. But with OCD, you need higher doses and for a longer period of time. And exposure and response prevention also takes around 2 to 3 months. So just to know whether it's helping, it can be several months, which is hard for people because they're in pain.
Sure. So in order to start, addressing some of these limitations, you've spent most of your career actually focused on trying to first understand the mechanisms of how some of these medications work, as well as, pioneering work into some new therapeutics in OCD. And I believe you did the first study of ketamine injections or infusions into OCD patients. And what did you see?
So we did the first study of just, let's see, giving somebody a dose of ketamine, if it can be helpful for them. And this was based on work that glutamate modulators in OCD had been having some promise, and an animal model called SAPAP3, where disruptions in glutamate protein that's important for scaffolding is when it's disrupted, you have these repetitive grooming behaviors. And so those two pieces of evidence together, we thought, let's test it. And after the single dose of infusion, there was a dramatic decrease within hours. And the first patient was like, this is like a vacation from my OCD. And I almost knocked me off my chair. It was so exciting that something that has a sort of rational reason why it could work actually works. In clinical research, that's unheard of. So, we basically said, let's see if this is placebo. Let's see if this is actually a real signal. So then we went on to do a randomized small pilot phase 2 study looking at ketamine from midazolam. We repeated that the saline and ketamine those individuals with ketamine had significantly more decrease in OCD than the saline, which is pretty flat line. And then we just finished an R01 study looking, replicating and extending the study in a larger sample and looking out for longer, and found that with an active control called midazolam, which makes people feel a little woozy, it's a little bit of an anesthetic like ketamine, we got, again, significant separation with a single low-dose infusion of ketamine out to 3 weeks, but not 4 weeks. So that was really exciting and heartening to see a story of progressively more rigorous controls and having replication. That's nice to see in science.
Yeah. So what do we actually know about what ketamine is doing in the brain and how it might be having some of these effects? I'm interested also that it's having such rapid actions, but also somewhat long-lasting effects as well. Do we understand how that's working?
Yeah, I think, well, fortunately there are a lot of labs that are looking into this and the molecular mechanisms of how ketamine working in animal models. Connor Liston's group, for example, is doing, very beautiful work at the molecular level. Then, there are people who are studying it at magnetic resonance spectroscopy, like how changes in the brain are changing levels of neurochemicals in a specific area of the brain. And then there are folks who are studying it with neural circuits at the fMRI level, as well as EEG and other levels. And so what we wanted to do in our study with OCD is to try and answer these questions in one individual with OCD while we're giving them a ketamine infusion, have them in the scanner, look at cognitive control circuits, so the ability to stop and start thoughts, and also look at, with magnetic resonance spectroscopy, do changes of glutamate and GABA change. So stay tuned. We're analyzing the data, but we're really excited to have the top line clinical data to show that some people respond and some people don't. And now we have the data and the tools in hand to be able to ask, are there any markers in the brain at baseline that would predict who goes on to response? And also to look at mediators to see what is changing in the brain that is associated with symptom change. Because that's really, we want to use ketamine as a tool to understand the fundamentals.
Yeah, not only, for its potential as an incredible therapeutic, but also to help us understand actually the disease process and make guided decisions about treatment as well. So one other question, ketamine has also, of course, had, tremendous potential as an antidepressant. I'm just personally curious about its role in OCD versus depression. Do we think that it's working through similar mechanisms across these various disorders?
I would love to know that. I'm so curious about that. We had sort of an early sense with a study looking at magnetic resonance spectroscopy, where we did it in OCD, and Matthew Milek at Columbia did a study, same exact design, same scanner, same everything, except the patients were depression. And in our study, GABA increased 1 hour post-infusion, which is an inhibitory neurotransmitter. And in their data, they saw glutamate, GLX, which is glutamate and glutamine, and GABA go up together. So it made me really think that there was maybe a different signature, different chemical pathway, different things that are happening in the brain that could be responsible for its effects. But I think more to come. I think I'm excited about that.
I can't wait to hear more about that. Yeah. So what are sort of the challenges in bringing a drug like ketamine to actual, mainstream clinical use?
Right. Well, it's an interesting challenge because it's a little bit the cart before the horse, meaning that ketamine is already FDA approved. It's already used at much higher doses for anesthesia. And so the concern is, the safety monitoring and application to mental illness doesn't always have FDA approval. So ketamine is not FDA approved for OCD. Intranasal enantiomer S-ketamine is approved for treatment-resistant depression and other indications. But it's one of the challenges is that in the clinics it's for fee right now, and sometimes they'll put our studies on the website to say, hey, you should do this for OCD. So it feels like it's already something that is being translated, but yet ketamine is a drug of abuse. It's called Special K. We ensure in our research studies that we're carefully excluding people who have that risk factor. And we also, so far from what we've seen is that the effects are transient. And so our next branch of research is to see how we can help people get better and stay better. But in the ketamine clinics, you're getting repeated dosing, and we don't know the long-term effects of getting repeated dosing.
Sure. Yeah. So what is sort of the next step for your research in looking at some of these novel therapeutics in OCD?
So we have a couple avenues of research. So one is looking at the opioid mechanisms of ketamine and OCD. So when you give ketamine, people feel high, elated, euphoric. And so is ketamine working because of that? Is it working, through glutamate and GABA or other pathways? And so in order to study that, we can do a pharmacological manipulation where we randomize people to get naltrexone, which is a mu-opioid blocker, before getting ketamine or not. So you can see if, it's a very simple question, right? If you block the mu-opioid, do you still get the anti-obsessional effects? If so, then maybe it's not important. But if it doesn't block, then the opposite is the case. So it's a very simple kind of toggle on the switch to see if it is important. And that will help us define whether we should invest more in glutamate, the glutamate mechanisms, MDMA modulation of the drug, or opioid mechanisms, but then invest in non-addictive mechanisms. So those are the two pathways to go, and we're hoping that study will help us. So if you know of anybody who's interested, definitely email us at ocdresearch@stanford.edu. We'd love to partner with anybody who would like to participate in research with us.
Yeah, we'll definitely put those links in the podcast, and we encourage anyone who knows anyone who is interested in any clinical studies or learning more about Dr. Rodriguez's research to definitely check out those resources. So before we let you go, unfortunately this has to be a short podcast, but before we let you go, I did want to talk to you. My training is in basic neuroscience, so I study synapses and protein signaling, and I love hearing about this clinical research. I mean, this is why many of us actually entered into science, but I feel like there's this sort of tremendous gap between the things that I'm doing and clinical research. And I'm wondering sort of what your perspective is on that gap, and if there is something we can do either as researchers to sort of start to bridge that gap, or what is needed to really sort of connect these two areas of science.
Absolutely. Well, I really, I think it's the study of neuroscience is so beautiful, and there are people out there who are in the clinical world who want to get involved and partner with people, myself included, many other folks. And it's important just to find folks who are interested in that translational space, spend time together, learn each other's language. We were talking before that a definition of a circuit to somebody who's doing very basic research is going to be at a very different level than somebody who's talking about circuit in a neuroimaging context. And in one sphere, you can do very precise, very causal mechanisms, and another you can do associations. But the ability to translate back and forth will allow you to get the most out of both systems, like being able to ask causal questions and then using that to sort of narrow the field of focus for human studies, and then human studies can then be translated back into the animal model to refine those studies and make them even better to detecting what's happening at the human level. So I'm really excited for what's to come, and I thank you for the podcast and the opportunity to talk about these things. And you are making it happen to help us form bridges across clinical and basic. So thank you.
Well, thank you for that. Yeah, and seems like a really exciting time in neuropsychiatric research. So I'm just excited to see what happens, and it was wonderful to talk to you. Thank you so much.
Thank you. Appreciate it. Take care.
Thanks for watching today.
If you like this video podcast, make sure to subscribe to our YouTube channel for more cool neuroscience.